hippocampus, two areas that have been recently implicated in anxiety in mice. So the reason why we are studying this is because, as you all know, anxiety disorders are extremely prevalent, and as many as 20% of people will get some sort of anxiety disorder throughout their lifetimes. And that results in a huge emotional burden and also in a cost of tens of billions of dollars annually in the US. However, the pathophysiology of anxiety is not very well understood. And part of that is because there are very few reports on the activity of brain areas associated with anxiety. So, as a first step to characterize better the circuits required for anxiety, I studied activity in the prefrontal cortex and the hippocampus in mice, which are two areas that have been shown to be required for anxiety in mice. And recent evidence has shown that the hippocampus in mice has a functional differentiation between the dorsal and ventral poles, such that only the ventral pole appears to be required for anxiety. And hopefully, understanding better the circuitry involved in anxiety might open novel avenues for development of new treatments for these diseases. So here a quick background for the talk. I'll talk about how the ventral but not dorsal hippocampus as well as the prefrontal cortex appears to be involved in anxiety. And furthermore, the ventral hippocampus projects directly to the middle prefrontal cortex, suggesting that these regions may form a functional circuit that may act to guide behavior during anxiety. Then my results will be divided in two parts. In the first part, I'll discuss how prefrontal cortical ventral hippocampal synchrony varies and is modulated during anxiety. And in the second part, I'll talk about how single units in the prefrontal cortex fire during exploration of an anxiogenic environment. So as I mentioned previously, many lines of evidence suggest that the dorsal and the ventral hippocampus have different functions. One such line of evidence comes from gene expression studies. For example, here we can see that wolframin is a gene that's expressed primarily in the dorsal hippocampus, whereas decorin is another gene that's expressed primarily in the ventral hippocampus. And many such genes have been found whose pattern of expression varies throughout the dorsal-ventral axis of the hippocampus. So this indicates that these subpoles of the hippocampus have distinct molecular identities, which are likely to have important functional consequences. Furthermore, differences between the dorsal and ventral hippocampus have also been found in physiology studies. To study hippocampal pyramidal cells in awake-behaving rats, recordings were obtained both from the dorsal hippocampus and from the ventral hippocampus, while rats foraged in a rectangular environment shown here. Here we can see the spatial firing rate maps of three distinct dorsal pyramidal neurons, and in each pixel, higher firing rates are indicated by warmer colors. So note that in all three cases, each of these neurons fire in a very restricted small area of the environment, which is called their place field. Conversely, in the ventral hippocampus, fewer cells appear to have place fields, and the ones that do have spatial selectivity have so in a much weaker way, with larger place fields and fuzzier boundaries. So both gene expression and physiology studies suggest that there are important differences between the dorsal and ventral poles of the hippocampus. Not surprisingly, differences between the dorsal and ventral hippocampus were also found in diverse behaviors. So for example, lesions of the dorsal hippocampus have been shown to impair working memory performance in mice, but lesions of the ventral hippocampus do not seem to have that effect. So although the ventral hippocampus does not appear to be required for working memory tasks, lesions of the ventral hippocampus do appear to impair behavior in innate anxiety tasks. These innate anxiety tests measure the avoidance that rats and mice have towards open, bright spaces and are not to be confused with fear conditioning, which is a form of learned fear that depends primarily on the amygdala. One commonly used anxiety paradigm in rats and mice is the elevated plus maze. This maze has two sublocations, the open arms and the closed arms. And generally, rodents have an aversion towards open locations. So for example, I'm sure every one of you have seen mice and rats in New York, and usually they never are in open, bright spaces. They always run around corners and dark locations. And that's presumably because rodents are more vulnerable to potential predators in open locations. So, accordingly, we can see that sham lesion rats explore primarily the closed region of the maze, similarly to mice that have lesions in the dorsal hippocampus. However, lesions in the ventral hippocampus appear to induce a partial loss of the aversion that these animals have towards the open spaces. So this indicates that the ventral hippocampus has an important role in anxiety in these paradigms. So the reason why these paradigms are thought to be important to understand better human anxiety is because, among other features, they have pharmacological validity. For example, drugs that decrease anxiety in humans, such as benzodiazepines, also decrease the aversion that rodents have towards the open spaces. So it's thought that because of this pharmacological similarity, both processes might have the same underlying mechanisms at some level. Furthermore, differences between the dorsal and ventral hippocampus were also found in their anatomy. So here we can see the basic output structures of the dorsal and ventral hippocampus. And the dorsal hippocampus projects primarily to the rhinal cortices, which are brain regions known to be involved in spatial navigation, which makes sense considering the dorsal hippocampus is involved in spatial working memory. The ventral hippocampus, on the other hand, projects primarily to these limbic regions, such as the amygdala and the entorhinal cortex, which is conducive to a role in mood regulation and fear and defensive-related behaviors. So, we know from the lesion studies that the ventral hippocampus is involved in anxiety, but it's not clear how the ventral hippocampus might act during anxiety. One possibility is that the ventral hippocampus, along with one or more of these downstream target regions, may form a circuit, and then the circuit may act to guide behavior during anxiety. We chose the medial prefrontal cortex as a potential member of the circuit for two reasons. The first is that it receives direct monosynaptic projections from the ventral hippocampus, and the second is that lesions of the prefrontal cortex also appear to increase exploration of the open arms in the elevated plasmates, similarly to lesions of the ventral hippocampus. So this shows that both the ventral hippocampus and the prefrontal cortex are required for the same anxiety-related behaviors in mice and rats. However, what can one do to study the potential interactions that these structures might have during behavior? One way to do that is to study their local field potentials. So local field potentials are large extracellular voltage fluctuations which arise due to synchronized synaptic input. And stratified areas such as the hippocampus favor the occurrences of very large local field potentials. Here we can see an example trace of dorsal hippocampal local field potentials in an awake behaving mouse. So note that there are many types of oscillations going on, but the most prominent oscillation is this one that happens at around 4 times half a second or 8 hertz. So this frequency oscillation that goes from 4 to 12 hertz is called the theta oscillation, and it is the most prominent and robust oscillation that's observable in the mammalian hippocampus. Here we can see the theta component filtered for this trace, and you can see that it does indeed carry a lot of the total energy of the signal. However, there are also other components to this field potential. So note that there are these faster oscillations here that happen around 5 or 6 times in each half theta cycle, and that's called the gamma oscillation. But it's responsible for much of the total up and down of the signal. So this is why in the power spectrum of this trace we can see that the theta region peak is the highest, whereas the gamma region peak, which is around 30 hertz, is lower. So the power spectrum is just a way to compare the relative amplitudes or power of different frequency components of the signal. Local field potential analysis can also be used to measure synchrony across brain regions. For example, it is known for a while now that prefrontal cortical spikes are commonly phase-locked to dorsal hippocampal theta oscillations, which are shown here. These spikes are simultaneously recorded with this dorsal hippocampal local field potential, and note that the spikes tend to occur near the peak of the oscillation. So there are certain phases of the oscillation in which the spike is more likely to occur. Thus, the spike is said to be phase-locked to the oscillation. One way to better visualize phase locking is by plotting a phase distribution histogram of this spike train, where percentage of spikes is plotted on the y-axis and the hippocampal theta phase bins are plotted on the x-axis. So note that there are some phases of theta at which the spikes are more likely to occur. This is why this spike is thought to be phase locked. And phase locking is a measure that could be used to measure the synchrony between the ventral hippocampus and the prefrontal cortex during anxiety. However, there are no reports on how the ventral hippocampus may synchronize the prefrontal cortex during anxiety. But what we do know from previous studies is how the dorsal hippocampus synchronizes with the prefrontal cortex during working memory, which is a behavior that's thought to require both these regions. To study this issue, recordings are obtained from the dorsal hippocampus and from the prefrontal cortex, while mice performed this working memory task called the T-maze. This task has 3 phases. In the first phase, the mouse is forced to go to one of the arms. Then, after a short delay period, the mouse chooses which arm to go to get the reward. And the reward will be in the opposite arm, not the one visited, but the other one. So note that during the center arm run of the choice phase, the mouse must hold in its working memory the location of the rewarded arm. And intriguingly, it was found that during the choice period of the task, phase locking strength of prefrontal cortical units were higher compared to the sample phase of the task. So this shows that theta range synchrony between the hippocampus and other downstream target regions can be modulated by behavioral demands. So now I'll go to the first part of the talk, which is on how prefrontal cortical ventral hippocampal synchrony can vary during anxiety. So what I've said so far is that the prefrontal cortex, the ventral hippocampus, but not the dorsal hippocampus, are involved in anxiety. And then the ventral hippocampus projects directly to the prefrontal cortex, suggesting that these regions may form a circuit. And furthermore, it has been shown that synchrony between the hippocampus and other downstream target regions can be modulated behaviorally, particularly in the theta range. So from this, we generate 3 hypotheses. One is that the prefrontal cortex should be more synchronized to the ventral rather than the dorsal hippocampus because it's only directly connected to the ventral hippocampus. The second hypothesis is that prefrontal cortical prefrontal cortical ventral hippocampal synchrony in the theta range may be modulated during anxiety. And third, there may be physiological correlates of anxiety in the activity of these regions that are thought to be required for anxiety. To study these issues, we implanted mice with electrodes in 3 locations, the prefrontal cortex and the dorsal and ventral poles of the hippocampus. And these lesions show that we could successfully target all of these regions. And this is a picture of how the mouse looks like with electrode implant. So following surgery and recovery, mice were exposed for 4 days in a small rectangular box that's kept in the dark. This box will be referred to as the familiar environment. This environment is thought to not be anxiogenic because there are no open spaces or bright lights. So it's a closed, small location. Presumably, mice are not anxious in this location, especially considering that they have been exposed to it for 4 days. Then, following familiarization, they're exposed to the elevated plus maze, which is the anxiety-provoking environment I mentioned previously. After that, following a rest period of 2 days, mice were exposed to the open field. The open field is an anxiety-related paradigm similar to the elevated plus maze. But, however, in this paradigm, mice and rats tend to avoid the center of the open field because this is a location which is aversive and open and brightly lit. So, first I'll show how the local field potentials and the synchrony between these regions looks like during exploration of the familiar environment, and later I'll show how these results and the synchrony between